Chapter 2 is a very long chapter. It's intended to be a review of things that you've taken before. But this letting you know, this is traditionally the content of a Chapter 2 in any A&P course. Depending on where you take this course, sometimes Chapter 2 is skipped if you have a requirement for chemistry before this. For example, when I was at Lemoyne, this chapter was skipped because this course would normally be taken after chemistry. But here we do include it. But this is the explanation as to why this chapter is as long as it is. But it's very important to understand the context of everything else, as we spoke about in Chapter 1. In order to understand how the body works, you really need to have a fundamental understanding of some chemical principles and the molecules involved. So that's why we need to go over this. All right. Try your best not to be intimidated by it, and make sure you use the study guide to keep you grounded, and understand that this isn't just one lecture. This would be, the way I do it, 3 or 4 lectures. So, pace yourself with this. And I understand that many of you might not remember all of these things. Some of you will remember more, and others won't. But so don't panic. Take your time. If you need help, make sure you come and see me. All right. So your learning outcomes, as listed in the syllabus, are here. This is sort of a rudimentary study guide, as you know, but use the study guide that I use for the exam to let you really know what you should focus on. All right. So we start off with the elements of the human body, right? So this is, as I mentioned, this is why we need to understand some chemistry. And this is divided out into major, lesser, or trace elements. And some of them, the major ones, should be obvious to you, right? Oxygen, carbon, hydrogen. You might not think of nitrogen. You will think of calcium, because you know that you need that in your diet. But you might not think of phosphorus, depending on how into nutrition you are, all right? Lesser ones would be sulfur, potassium, as you can see, and so on. And you'll see why as we go through this course, all right? Lesser means that doesn't mean we can live without them. We need them, all right? So even trace, if you look here, trace elements, chromium, cobalt, copper, fluorine. So here you can see them again. I prefer to look at them this way because you can see the chemical symbol. I know you don't need to be a chemist to do this, but you'll know your book will refer to it using the chemical symbol or the elemental symbol. So try to learn these, all right? So at least learn those, the ones in this bigger box, all right? Make sure you know these. The other things that I just want you to notice, there's a few more trace elements listed here that you might find surprising, like silicon and things like that, right? So, but there are quite a few, okay? And this percentage here is based by body mass, and that's, notice, including the water. All right, just to keep you grounded, but you should be able to identify definitely these 4 as your major ones, okay? Okay. Now, let's get into this. So when we're talking about these elements, all right, so when we're talking about atoms of the elements, the atom itself has the 2 major parts, the nucleus and the outer shells or orbitals. We're going to go over what those orbitals are rudimentarily, and why they're important, because they're really important. And in A&P, we don't really talk too much about the nuclear parts, just other than knowing what's there. If you take chemistry, then you will learn some nuclear chemistry as well, which is quite interesting. But anyway, so in the nucleus, we have our protons. They have mass, meaning that, relatively to, we're going to talk about electrons in a sec. So protons are quite large, and they are combined with neutrons in the nucleus. So the neutrons are neutral. That's why they have no charge, all right? So they're neutral. Protons have a positive charge, and both of them have mass. The mass is very similar to each other, okay? And then on the outside, we have the electrons. They have relatively no discernible mass. That doesn't mean that they don't have mass, right? Because if you know the definition of matter, which we'll talk about, is something that has mass and takes up space, but obviously they are part of matter. So they have a mass, but relatively speaking to the protons and the neutrons, the mass that they contribute to the atom is negligible, all right? But they have a negative charge, all right? So the electrons have a negative charge. And then here's your sort of rudimentary drawing of a very basic atom. And the reason very often these electrons are shown as a cloud is this is actually just this orbital is actually where the electrons would exist most of the time. So it's like a probability thing. So really they think of it as a cloud of electrons, all right? Now, when it comes to this, so we have our atomic numbers. If you look at a periodic table, the smaller number is the atomic number, and you'll notice it's in order, you know, 1 all the way up to a very large number. And that is the number of protons. It's always the same for any element, all right, is the atomic number. That will always be the same. And here you can see the atomic number for helium is 2, meaning that it has 2 protons in it, okay? Now, the atomic mass, or the mass number, is roughly equal to the number of protons plus the number of neutrons. So that's the bigger number. And here you can see the mass number for helium in this case is 4, right? So you know you have 2 protons, and then your atomic mass is 4. So then you can do some basic math, and you can see that you have 2 protons and 2 neutrons in the nucleus of helium, all right? This chart here is just to show you the relative mass. So, you know, if they would, and mass unit, which is arbitrary, but if they assign 1 to protons and neutrons, because the mass is relatively the same, it's not exactly 1, but they assign it 1. Relatively speaking, the electron is 1/1837th of that, okay? That's why they say that the mass of an electron is negligible, doesn't add much to the actual mass number, okay? So that's why we always just consider the neutrons and the protons, okay? And here's your chemical symbol or elemental symbol that goes with helium, all right? All right, now, the important thing about this atomic mass, all right, is that not all of the atoms of an element have the same mass. I did say that any element will always have the same atomic number, so it always has the same number of protons. But different forms of the same element, which we call isotopes, okay? Isotopes have the same element, have the same number of protons, but a different number of neutrons. And because of that, they have a different mass, all right? So if you look at hydrogen, for example, normally what we think of when we think of hydrogen is this one here, this 1 here, actually just a proton and an electron in that atom. But we also have deuterium and we have tritium, all right? So deuterium has 1 neutron and tritium has 2 neutrons. And they're all hydrogen, but they're different isotopes of hydrogen. Now, I don't want you to be confused with radioisotopes. Radioisotopes are unstable isotopes. Just because it's an isotope doesn't mean it is unstable. So radioactive isotopes give off energy because they're unstable, and then they revert to another type of element because they lose that energy. You know, that's a little bit more advanced than what you need to know. But this is how they can date fossils. They use diagnostic imaging and cancer treatment. They use radioisotopes because of this energy that's given off. It's very useful because you can actually know how long it takes for that energy to come off. Now, for example, when we're looking at deuterium and over here, hydrogen and carbon, carbon also has 3 different isotopes. This one here in hydrogen is considered radioactive. All right, so tritium. It's not radioactive in the form that, oh my God, it's going to kill me, because there's different types of radiation, but it is unstable, so it's considered a radioisotope. And then when it comes to isotopes of carbon, it's carbon-14 that's considered the radioactive one. Now, carbon-14, the type of energy it gives off, is a little bit more dangerous than this one here. All right, so this gives off what we call beta emission. This gives off alpha. And then there's other ones like iodine. Its radioisotope is really hot. It gives off gamma radiation, which is dangerous. Not that carbon-14 isn't dangerous, but you can block it with plastic. If you're working with hot iodine, you need lead. All right, just to give you an example about even when something is a radioisotope, there's different degrees of danger and precautions that you need to take, but not all isotopes are radioisotopes. Okay. It's probably more than you need to know, but I'd like you to make sure you understand. Now, we also have these things called free radicals, and you've probably heard of those guys. One of the reasons in medicine we think about free radicals, the only thing to counteract free radicals are your antioxidants. So you could think of pomegranate juice. That used to be one that was quite popular. But the reason they wanted you to have that in your diet is because what that antioxidant does is that it neutralizes these things called free radicals, which can be damaging to the body. Okay, so a free radical is when you encounter a molecule, all right? This is not an element now. We have a molecule. It's made up of nitrogen and 2 oxygens, all right? And you have an odd number of electrons, right? So here you have this 1 electron sitting here. Notice it's not in a pair. We'll talk a little bit about that in a little bit, but a free radical is when you have this unpaired electron, and it makes it kind of reactive because it wants to pair up with another electron. We'll talk more about it in a few minutes when we do bonding. But this makes these free radicals kind of damaging to the body because they'll steal electrons from other things in your body and damage things. So if it damages, I say things, let's say DNA, for example, right? You can take an electron from one of the parts of your DNA, and that could cause a mutation that could be damaging. All right, so that kind of thing. All right. So what we want to use are these things called antioxidants, as I mentioned, like pomegranate juice. It has antioxidants in it. And basically, what does that do? Well, you have an antioxidant, that is a molecule that can donate an electron to neutralize this free radical, and it just makes things safe. So antioxidants neutralize unstable free radicals. All right. So in terms of a diet perspective, hopefully that is helpful, so you can kind of understand what a free radical is. Okay, now we need to talk a little bit about ions, because we're going to get into bonding. All right, and we'll do that shortly, but I want to sort of ease us into that. Okay, because you've probably heard of positively and negatively charged ions, and you have them in your body, right? So when we're thinking about these elements in the body, and you'll see this very quickly. Very often we're not talking about elemental calcium, for example. We're going to talk about a calcium ion, all right, or the potassium ion, or the chloride ion, all right. Now, what has happened here is that, let's go here now, is that what we have actually is a charged form of our elements, okay? And it doesn't have to be an element. We've got charged molecules as well, but we're keeping it, a little more straightforward here. So sodium and chloride, and if you know some chemistry, you'll know that they come together, make salt, sodium chloride. We'll talk about that as well when we do bonding. But what I want you to understand is if you have an element or an atom that is missing an electron that it should have, or missing multiple electrons depending on the charge, you have something that's positively charged because you've lost some electrons which are negatively charged. So that makes our element or our atom now a positively charged, and we say it's a cation, all right? So cations are positively charged. And then conversely, you can have an atom that has taken on extra electrons, and that would then give it a negative charge, and that is called the anion, all right? And this is pretty important when we talk about bonding, which we'll get to. So cations and anions attract each other. And elements that typically, or molecules that typically form ions are called salts, right? So our classic salt, table salt, sodium chloride is obviously an example of that. So sodium chloride in the body doesn't exist as sodium chloride. It breaks down in the water, which is why we'll talk about it as well. And it exists as its ions in the body, and which is really, really important for our chemistry. all right? What are these things called? Well, they're actually called electrolytes. So I'll get to that in a sec. All right, so now, when we're talking about how these salts dissolve in our body, right? So we could think of our body as mostly in terms of what we have as a solution is aqueous. So we have aqueous or water-based solutions, and a solution is a mixture of two or more substances, right? So usually there's one that's more and then the other. So the solute is the thing that's going to disperse, or the thing is that is dispersed in the solution, and the solvent is the thing that's doing the dissolving. So it's usually something that's greater in quantity, all right? Now, in human chemistry, obviously, and hopefully this is obvious, our solvent typically is water, okay? And water is hugely important because of that. But it has some very interesting chemistry, which we'll discuss. And anything like our salts that is dissolved in it is called the solute, all right? So we're sort of like a bag of salty stuff is basically what we are, all right? Now, we have a special name for those ions, or salts that are dissolved in that water, and we call them, as I mentioned before, we call them electrolytes. Now, it's kind of weird. Why do we call them electrolytes? And this is going back really to chemistry, and you know that when you have like Gatorade or something like that, there's electrolytes in it, but you probably didn't really stop to think about, well, maybe you did. Think about what does that mean? Well, it just means that there's salts in there that dissolve into their component ions. Okay, so a salt doesn't have to be sodium chloride. It could be potassium chloride or other types of ions that form different kinds of salts, right? So that's why I said I qualified sodium chloride as table salt. And indeed, that is a very important one in the body, right? Now, why are they called electrolytes? And that's because if you connect them with a wire, or you can collect electrodes with a wire in an electrolyte, it will conduct a charge, right? Conduct electricity. And this is why things like ECGs work and things like that you'll talk about in AMP2. Actually, we'll talk about it in this one, AMP1. We'll get to that. I was thinking about it in terms of the lab, because you do the ECG lab in semester 2. But we will be talking about how these salts carry a charge in the body in a little bit, but you'll talk about it a lot in AMP2. Okay, so you do conduct charge because you are full of electrolytes, all right? But this is where this name comes from, and that's why they're called electrolytes. Now, and it just occurred to me that I haven't boosted this up, so let's just make this bigger. Okay, now, electrolyte balance is really, really important to the body, all right? So you need to have the correct amounts of these different electrolytes, which is why we have things like Gatorade, right? We have things like Gatorade to help us after we've lost a lot of water, we sweat, and we want to get our electrolytes back in balance, all right? So this is quite important. We're going to stress that as we go through, and as you go through AMP, you'll learn why the amounts of these different ions in your salts are important to the functioning of the body, all right? So it's important for, and I listed here, chemical reactivity, osmotic effects, and the electrical excitability of nerves and muscles. Very, very important. So if you have an imbalance, it can lead to coma or cardiac arrest, as I've listed here, all right? So here are some of the main ones, right? We already talked about sodium chloride, right, down here. But we also have things like calcium chloride, disodium phosphate. Now this might look really weird because this is an ion. It's a multi elemental ion here. Okay, so sodium and phosphate, magnesium chloride, potassium chloride, sodium bicarbonate. This is another one that stays as an ion. We'll talk about it as well. And of course, sodium chloride, like I mentioned before. So these are your main electrolytes, and this is what they become when they're in your body, and you use these, right? And this one's really important. We'll get to that very shortly, okay? Right, just looking at my notes because I want to make sure I got, all right. And I mentioned the ECG. I'm on page 3 of my notes. All right, now, and here, if you look in the body, for example, about how important these different ions are that compose these electrolytes, notice that they don't have the same concentrations in different compartments of the body. And that is hugely important. We call these things, these differences in concentration across barriers, we call these gradients. All right. You don't want to reach equilibrium because that would cause death. All right. We need these gradients to allow the body to function. So for example, this barrier in between the different compartments that I'm showing you here is actually the cell membrane. All right. So if you look at the concentration of sodium in the extracellular fluid, or the fluid around the cell, all right, and that is often thought of as the interstitial fluid, all right, versus the intracellular fluid, so that's the fluid that's inside the cell. Notice there's a huge difference in concentration, and we'll talk about how that works, or how we maintain that, okay? Same thing with chloride and potassium, all right? So these are really, really important gradients that we have to maintain. And that is important to keep our electrochemistry functioning, and that keeps us functioning, all right? Now, in terms of the chemistry that's taking place here, now if we look at our atom, all right, now this is kind of a fundamental thing. We're going back to our basic chemistry. And here we have just a little portion of the periodic table, right? So we have hydrogen and helium at the top. And if you have like a full one, you can look at that, or you can Google one and look at it. The second row, all right, notice what's happening. So these electrons that are in the outermost, or in the, I'm getting ahead of myself, that are in the orbitals around the nucleus are placed sequentially into these shells, or these orbitals. So notice in hydrogen you've got 1, helium you've got 2 electrons, which is represented by these little yellow dots. And in the first row of the periodic table, you only have 1 shell. Now, when you get to the second and the third shell, which is where most of our chemistry that's taking place in the human body is happening, all right, our bonding, which we're going to get to, we're dealing with the second and the third shells. And something I want you to notice is that with the second and the third shells that we're dealing with in this part of the periodic table, our shell, our outermost shell, is full. So you can see as we go across the periodic table, we're adding more electrons. It fills up with 8 electrons. Here in the first shell, there's only 2, and then you're full. The second and the third, we fill up that outermost shell with 8 electrons, okay? So we call this the octet rule, and it applies to not all chemistry, but chemistry that is driving a lot of the biochemistry, the human chemistry that we're dealing with in A&P. So it's nice to think of the octet rule. And what do I mean by that? Well, the element, or the atom, would like to feel full, all right? It would like to have a very stable outermost shell, all right? And it's stable if it's full. And this desire to get to fullness, it drives a lot of the chemistry, all right? So and I'm going to show you how this works very shortly. So we call this the octet rule. So these elements would like to have 8, because then they would be full and they'd be very, very stable. And if you know anything about your chemistry, you know that over here on the periodic table, in the column that's at the far right, these are what we call noble gases, and they're very, very unreactive. And the reason they're very unreactive is because their outermost shell is full. So they're very stable, all right? So all of the other positionings here are jealous in a way. They want to be like these guys and be very full and happy, all right? And that, I know that sounds silly, but it's a good way to think about, good way to think about it when you're trying to figure out how this chemistry is working, okay? So let's talk about these bonds, all right? So the outermost shell, so if you look here, the outermost shell is called the valence shell, all right? It seems like here the outermost shell is only 2, but in neon, the outermost shell is this octet here, and in argon, it actually is up 1 level here. The outermost shell is there, and that outermost shell is called the valence shell, all right? And that valence shell is the one that drives the chemistry that's taking place with these, between these different atoms, okay? So in a chemical reaction, we say that these valence electrons can be gained lost, or shared. And the driver then is to reach the octet rule, all right? So now, page 4 of my notes, all right? So we're looking at this idea of bonding, all right? So the one thing to think about here, and this gets a little bit into the periodic table, and you can see on my notes on page 4, on the left side of the periodic table, and I'll go to the rudimentary one here. This side, these guys are called the metals. So these first 2 columns, metals. And over here we see nonmetals, right? And as you go across the periodic table, you're getting less and less metal-like, all right? The next part of the periodic table is this really weird, funky area that's called the transition metals. So, but we're not going to talk about that here. But in terms of the chemistry that we have talking about here in A&P, it's a good way to think about it are metals and nonmetals, all right? So when you're looking at this, metals, all right, so those things that are on the left side of the periodic table tend to want to lose electrons so they can get closer to their closest noble gas. Whereas nonmetals will tend to want to gain electrons so that they can also become closest to their, similar to their closest noble gas, all right? So the metals will lose electrons and become cations, and the nonmetals will gain electrons and become anions, all right? So I have hydrochloric acid basically here, HCl. So if we look here, we'll go back to our periodic table. If you look at chlorine, all right, it's right here. It's got 7 valence electrons. So it would love to just take 1 more electron and it would become like its consequent noble gas, which would be argon, right? It still would be chlorine because of the number of protons it's got. But it would be behaving like argon if it just took 1 more electron. So it would really, really like to do that, right? Whereas hydrogen, all right, it's perfectly willing to give up that electron. So, or these ones here, so think of lithium. This is probably better because we're dealing with an octet again. Lithium, right? So if it lost its 1 electron, it would be like its closest noble gas, which would be helium, right? That's probably an easier one to think of in terms of this example, all right? It would still be lithium because of the number of protons it would have, but it would be similar in a way to helium, but it would have a charge. It would have a charge of +1, so it's not helium, right? Same thing with the chlorine. It's still chlorine. It's just chloride ion and be, negative charge to it, so that it would be able to behave like argon if it could do that, right? Now, this is why chlorine and hydrogen like to react with each other, because they will then be stable in a way, right? So let's go forward to that spot there. All right, here. So the cation and the anion will interact with each other, and then as such, both of them will have a full valence shell. All right. So the classic example here, is hydrochloric acid or sodium chloride, which we're going to use to help you visualize this. All right. But understand that on one side of the periodic table, if they lose some electrons, they'll be happier. And in the other side, if they gain some electrons, they'll be happier. All right. Not that atoms have emotions, but hopefully that makes sense to you. All right. And this is what's driving the bonding chemistry that's taking place. So let's go over this. So we have different types of bonds that occur. All right. So the first types of bonds that I'm going to talk about are what we say are intramolecular bonds. Now these, when I say intramolecular, so I-N-T-R-A, intra, meaning within a molecule, all right? So this is making a molecule. One type we have are covalent bonds. So covalent bonds are a situation where you're sharing these electrons, and these are really, really strong bonds, all right? And you can share them different ways. You can share those electrons equally, or not equally, all right? So when we say that we're sharing our electrons equally, right, and this ends up being atoms that are the same, right? Otherwise, it wouldn't be equal, right? So there, this is when we have like hydrogen gas, oxygen gas would be a sharing of equally between 2 different atoms, okay? And like I said, this is a very, very strong covalent bond, and we say it's non-polar. All right. The reason we say, and I'm going to get into the polarity more when we get into polar covalent bonds, it'll be easier for you to see. But if you look at a non-polar covalent bond, we're sharing those electrons within a molecule equally, and so that the molecule is sort of symmetrical. If you look at it that way, there's nothing, to say that one side is different than the other side. These electrons are zipping around here, and they're being shared nicely between the two atoms. Okay, and as such, then they both have a full valence shell. They're very happy like this. Okay, but we can also share unequally. All right, so if you have something like, in this case, water, one side, so if we look at water, and we're going to talk a lot about water, if you look at oxygen and hydrogen, now let's go back to our periodic table, right? So oxygen, if you notice, has quite a few electrons around it, right? So in its outermost shell, it's got 6, and then it plus has these 2. So there's 8 electrons total around oxygen. So it's a kind of negative, right? It's not negative in terms of a charge, but it's more electron dense than, say, hydrogen, right? So this molecule would, even though we're covalent, we're not stealing electrons in this case, we're sharing them, but the electrons tend to be closer to the oxygen because it really would like to be like neon, right? So this is kind of what's happening here. They're pulling those electrons a little bit closer. They're still being shared, all right? They're being shared, but oxygen he's a bit of a bully. He wants to have a little bit more of the electron than the hydrogen does. And the hydrogen is okay with that because, he's fine with doing that because he's kind of happy, being stable. And it'd be more stable if you kind of gave up that electron, to the oxygen in a way, okay? So if we look at it, what we end up having is a sidedness to this, right? So we call the sidedness, and one side is, it doesn't have a charge, but we say it's slightly negative, so delta negative, and the other side is delta or slightly positive. And because of this, it's kind of like a magnet, and you have poles on a magnet. So this is why we call this a polar covalent bond, because the electrons are being shared, but not equally. So they're being shared unequally. And when you have a molecule in there, you have this intramolecular forces and this polar covalent bond, it creates a molecule that has poles on it. And water is a really good example. There's other ones as well. All right, so it's not that there's a real charge on this thing is neutral, but there's a difference in the placement of the electrons, so that because there's more electrons on one side, it's not negatively charged, but it's inherently more negative because electrons are negative. Okay, so I hope that makes sense to you. Okay, so that's your polar covalent bond. Now this is different from the ionic bond. So this is where we were talking about when we have electrolytes, right? So here we're not sharing electrons, all right? We're actually donating or gaining, all right? And again, let's use our salt, our table salt as an example, right? So here you really have a non-metal and a metal reacting with each other. And that sodium, and we'll go back here, right? So sodium, in this case, and this is an ionic bond, right? So did I go past it while I was talking? I did. I'm sitting here talking away and then forgetting what I'm doing. There we go. Sodium, right? Really would be happy to get rid of this little electron here. It'd be like neon. And then chloride, of course, would love to take it, be like argon. Okay, and here we're not sharing those electrons equally. We are literally, chlorine is going to take it. It's going to become negatively charged ion. Sodium is going to give it away, and it's going to be positive. All right. Now this might be surprising to you, but these ionic bonds are actually quite weak bonds, even though there's this like charge between them. All right. So remember here we have positive charge ion is the cation, negative is the anion. All right. So here you can see the electron being pulled by the chlorine, and the chlorine then taking that on. And as such, we have a positive charge on the sodium, a negative charge on the chloride, right? They each have a full valence shell, both happy, all right? And they make a salt, all right? And this, of course, since it is a weak bond, it's very easily to dissolve in water, and that becomes our electrolyte, right? But that would be your ionic bond. Let me just see if I have anything else in my notes. All right. Now, the next type of bonding, which is really, really important in the body, is what we call hydrogen bonding. okay? Now, hydrogen bonding is not an intramolecular bond, all right? This is an intermolecular bond. An intermolecular bond means that it's taking place between molecules, all right? So between molecules of water, or in this case on the slide, between a molecule of water and a molecule of ammonia, okay? So this is why we say it's intermolecular, all right? Hydrogen bonds, a very special kind of bond is that they always involve hydrogen, all right? So it's between molecules that are polar. So notice ammonia, and you know water, we just talked about that. These are polar, all right? So they're between polar molecules. But in this case, in a hydrogen bond, hydrogen is bonded to an electronegative atom, all right? And that is typically oxygen, nitrogen, fluorine, all right? Sulfur and chloride as well, all right? But you're mostly going to see it as oxygen, nitrogen, or fluorine, or you know, in the context of what we're dealing with, okay? So the examples are, so here you have a very electronegative, so one has a lot of electrons bonded to a hydrogen, and that's going to then, so here's your sort of positive side, right? Because the, you know, the electrons are being pulled to the fluorine, and that will then interact with the fluorine on another molecule in this case, all right? Or here, so imagine this is OH from the hydrogen, OH, and you know that this is more sort of negative, not negatively charged, but it's more electronegative on this side than it is here. So this sort of positive, and it can react with the more sort of negative-ish nitrogen on the ammonia, all right? And that's what you're seeing here, all right? And this is called a hydrogen bond, all right? It is a weak force between polar molecules, but it's so important to the body, which is why we have a whole little blurb on water in this unit, okay? Now, the next thing is what we call van der Waals interactions, all right? And van der Waals interactions exist because of the different placement or the different distributions of electrons around a molecule. These van der Waals is, again, an intermolecular bond. Sometimes this is called London dispersion forces, right? I put that in my notes, okay? I don't want you to get confused by that, right? So van der Waals and London dispersion forces are the same thing. And they exist because the electrons, remember, they're not static. They're moving around the different molecules. And as such, they're creating like sort of areas that are a little bit more, not charged positive, but more positive, and others are a little bit more negative. Very much like we talked about with hydrogen bonding, right? In terms of why these are interacting like magnets, all right? And lo and behold, hydrogen bonds are actually a very special kind of van der Waals interaction, all right? But they're so important that we give them their own category, all right? But you can imagine that van der Waals, like these kinds of interactions that are taking place with things that don't involve hydrogen and all of that other stuff, right? That isn't a hydrogen bond, okay? But still very important. They're considered weak, but still really important. So how weak what do I mean by weak? Well, they're strong enough to help a gecko climb up a wall, right? So those are van der Waals interactions that help geckos do that. And they're trying to use it in robotics, right? So here's just like a sort of like a plastic pad that they're using to pick up this glassware, chemical glassware. This is actually looks pretty heavy, right, compared to this little pad. But this is van der Waals at work, right? So van der Waals interactions, even though they're so weird to understand, they're really quite important, even though they're still considered a weak bond. But when you have a whole bunch of them working together, really quite important, all right? Okay, so hopefully that makes some sense, all right? So this is the important chemistry. Now that would be one lecture. Now, so if you're following along, you could stop here that would be lecture 1. So let's go into lecture 2, water. All right, so we just talked about water being the main solvent in the body. We know that water is capable of doing hydrogen bonding, which are very special London dispersion or van der Waals interactions. Water molecules will interact using H bonding between each other, as you can see here, okay? And remember, 60% of your body weight is water. And in the blood, you've got, I think it's like 98% water, okay? So water is huge for your chemistry, right? Not only as a solvent, but in terms of the properties that water has, all right? So I'm going to mention quite quickly here the different properties of water. So if you look at it, right, you probably know this, water expands on freezing. That's why ice floats, all right? And basically when those hydrogen bonds arrange themselves in the solid, the spaces between the water molecules is larger than when they're in the aqueous or the liquid form. And that's why liquid water is more dense than ice, and that's why the ice floats. But it takes an incredible amount of energy to break these bonds. Even though they're considered weak, they're very stable. They want to hang on to each other. So in order to boil water or to vaporize water, it takes a lot of energy to go in, all right? So I'm going to get into the importance of that in a sec. So water expands on freezing. That's number one. Now, I just mentioned to you, oh yeah, this is why that's important. Like a polar bear, they're lost without ice, right? And this is why we're so worried about them in the Arctic right now, all right? Just having a sip of my tea here. So they need to be on the ice floes to hunt, and because seals live in water, their favorite food, okay? So this is how they exist, and now with the ice disappearing, there's a problem, all right? And there's other things in the environment that are important for that ice floats. Now, second thing, I said to you that it takes a lot of energy input to vaporize water. Now, we say then that water has a high heat capacity, all right? So what that means, it has the ability to moderate temperature. And this is why in the summer, you want to go down to the beach, right? Because it's always cooler at the beach. And the reason it's cooler at the beach is because the water is able to absorb that heat. And it takes a long time to be saturated with energy, all right? So it tends to be cooler there in the summer. And then in the winter, it's slowly going to give off that heat. And that's why in the winter, it's warmer along the coast, right? So water has this ability to absorb and hold a large amount of heat energy. Now, this should make sense to you. This is why you sweat. Now, sweat is mostly water, right? So it's not all water and salt. But you deposit this liquid on your skin to help cool you off, because as it evaporates, it's going to absorb the energy or the heat from your body and cool you off, all right? So we call that evaporative cooling, all right? So really important in terms of keeping you at your proper temperature. The other thing is that water is cohesive, so it likes to stick to itself, right? So that's what the hydrogen bonding is really quite dramatic when you see it, this water spider. And you could do the same trick if you could get enough of your weight distributed around the water, okay? And I think I've seen some kind of funny videos of people trying to do this, but basically because water holds on to itself, we call it cohesive, and it will do this kind of thing, has these little divots. You see it in the lab with the meniscus, right? The meniscus likes to climb up or down the glassware, all right? That's why I drew it in my notes on page 7. This is why when you're reading volumes in the lab, you always read from the bottom of the meniscus, all right? I've drawn a positive meniscus in my notes, so it's curling upwards. And you look at it from the bottom of the meniscus, and that gives you the volume. And water tends to behave like this. There's other fluids that also will form a meniscus, but they'll do a negative meniscus, right? But here we're talking about water, okay? So a positive meniscus, and that's because the water molecules are crawling up the side of the glassware, and in so doing, they're pulling other molecules, and they make this meniscus. So it's cohesive, all right? It's also important for plants, right? So plants, they drink from their roots, yet the leaves need the water. So that water is pulled up the trunk to the leaves using the cohesiveness of water, right? So as water evaporates from the top, it pulls all the other molecules up with it. And that's how that works, all right? Yeah, and that's obviously important to us because we need plants, right? What to eat. The other one, water is an excellent solvent. We already talked about this. So remember, the solvent is the liquid or the medium in which other things dissolve, and the solute is the thing that's dissolved. And in the body, water is typically the solvent. And it's really good at dissolving other polar molecules as well as ionic molecules. It's not good at dissolving nonpolar things, which we'll talk about, all right? So if it's dissolving a salt, like sodium chloride, you can see that the negative parts of the water molecule orient themselves around the positives, and the positive parts of the water molecule will orient themselves around the negative, and in so doing, easily take this salt apart, okay? And it will dissolve in the water. As long as there are enough water molecules to do that, you'll have a salty solution, okay? Now, we say things that dissolve really well in water. We call them hydrophilic, right? So like I just said, polar molecules or ionic molecules, really good dissolving in water. So they're hydrophilic, hydro meaning water, philic meaning attracted to or water loving. Hydrophobic molecules don't dissolve very easily in water. And these would be things that are non-polar. It's nothing for the water molecule to arrange itself around to dissolve them, all right? So here we say they're hydro, meaning water, fearing, phobic, all right? So this is where we get this thing called like dissolves like in chemistry, right? So you need hydrophobic things to dissolve, you need a hydrophobic solvent to dissolve a hydrophobic solute. And you need a hydrophilic solvent to dissolve things that are hydrophilic, all right? And the classic thing is oil and water. So you already know that oil is going to be nonpolar, all right? Okay, hopefully that makes some sense to you, but it is an excellent solvent, but it can also easily dissolve really, really big molecules like proteins, which we'll talk about, because the water molecules can arrange themselves around the charges, and we'll talk about how that works. And proteins can dissolve even though they're really big, all right? Now, water is also really important because it participates in our chemical reactions that take place in the body, all right? So many of the chemical reactions that we have are ones that we call anabolic, or ones that are building things, or catabolic, or breaking down things, all right? So when you're doing an anabolic and you're making something from some units, you're gonna make bonds. And when you're doing that, you're gonna release water. So you're dehydrating the things that you're putting together. So we call that a dehydration synthesis. And the energy that's going in is going to be stored up in the bonds. And we're going to talk more about this as we go. Just keep it in mind. So you're going to add energy, you're going to release water, you're going to form stuff, anabolic. Then to break things down, you're going to have to add water, all right? And you're going to release energy. And that's called hydrolysis, hydro, water, lysis meaning breaking, all right? So you're going to break water molecules apart to break down things. And then as you break the bonds, you're going to release the energy, all right? And these things together, anabolism and catabolism, make up metabolism, all right? Which of course we're going to talk a lot about in this course. Now, you're also supposed to be, to understand how we make up different types of solutions in the body, right? So a solution are different types of mixtures in the body, right? So mixtures in water can be solutions, colloids, or suspensions, all right? So a solution, you have a very small amount of solute. The particles don't settle out, it's transparent, okay? A colloid, you've got middle-sized particles, it's kind of cloudy, but the particles still don't settle out, all right? I'm just trying to see, do I have examples in here? No, I don't. All right, suspensions, large particles, they do settle out. It's opaque. Blood would be a good example here. A colloid would be like milk. An emulsion is when you have two liquids that are mixed together, so oil and water. If you can, when you shake it, if you have a container of oil and water, you shake it really, really fast, and for a while they all look like they're in one solution in a way. It's not really a solution, it's an emulsion. And how you know you have an emulsion is that it will separate into two components. All right, so here you can see that here. Blood is sort of a suspension, I guess. And then here you have your emulsion, and this is sort of the different kind of things. So here you have milk, and here would be like salt water, okay? So hopefully that makes sense to you, and that's sort of what they're showing you here. Okay, so those are the different differences. And then there's a video here to help, right? I've got these videos that we'll go through. I don't have time to show the videos in an online course, so, but it's there, okay? Now, so the different types of mixtures, here's another table that will help you orient yourself. So different types of examples. So solution, glucose in blood, oxygen in water, that kind of thing, colloid proteins in blood, milk, like I said, suspension, blood cells, just sort of hanging out there, fats, that kind of thing. All right. So again, you can also say that a solution is dilute or concentrated. And this is kind of weird because you have to compare them between each other, but a dilute solution basically has a small amount of solute in a given solvent, and a concentrated solution has a large amount of solute in a given solvent. Basically, a solvent can only hold so much solute. And once you get too far, even if it's salt and water, there's just not enough water anymore to dissolve that salt and will come out of solution. So at that point, before it starts to come out of solution, you could say that that was a really concentrated solution of water, salt water. Versus much less, you could say it was dilute. But you kind of need to have it relative to each other so that you can really sort of imagine that. Then there's different ways of expressing concentration. You probably have seen in chemistry we use molarity. Molarity is that pretty much only chemists do that, or you use that in a science lab. You express concentration as molarity. And basically that's based on the mole, right? The mole being an arbitrary number of something, and just like there's 12 somethings in a dozen, right? So a mole is 6.02 times 10 to the 23 of something. I know that sounds weird, but in this course I don't explain where that has come from. But it's, as a chemist, when I see a mole, I know it's 6.02 times 10 to the 23. It's just much easier to think of it in terms of moles than in factors of that weird number. And molarity then is the number of moles that exist in a liter of solution. All right, so molarity is the way of expressing the concentration of that solution. But in practical speaking, out in the world, people don't do that. They usually do a percentage concentration. And I've listed some examples in my notes. You can do a percent per volume. Like say, for example, I have in my notes, I have 1 gram in 100 milliliters. That would be a 1% solution. So that we call a weight to volume solution. We could also do a percentage by mass. So the mass of a solute and a mass of a solution, so like 1 gram in 100 grams of solution would still be a 1% solution, but it would be a mass to mass percentage. And that's usually how the world works. Outside of the chemistry lab, it's usually how, even in the chemistry lab, when we get hydrochloric acid in the lab, it's a 38 mass solution, like so 38 grams of hydrochloric acid in every 100 grams of water or whatever it's made up in. All right, so 38% is pretty concentrated. But this is just an example. So there's different ways of expressing concentration. It's just something that you should be aware of. A lot of you are going into nursing, so you're going to see these different types of concentrations. Now, the other thing to be aware of are acids and bases. And that brings us to the pH scale, which is, ironically, seems to be such a confusing thing. And I'm going to try and break this down for you. So if you're not following what I'm saying right now, go back to my notes, have a look, all right? So you probably learned pH is the negative log of the hydrogen ion concentration, and that was it. And then they showed you that it's a scale between 0 and 14. It's above 7, it's basic. If it's below 7, it's acidic. If it's 7, then it's neutral. And that's all I told you. So let's take a little bit of the mystery away here. So we're here, like I said, that's basically what I just said, which is on this slide here, right? But what does it mean? And why is it so confusing? All right. And the reason that it's confusing is because things that are more acidic have a smaller number, right? If it's below 7, it's more acidic. Why is that, right? So, and it's because of this weird math. And it's just because chemists like to simplify things, just like, a mole, right? So now let's take this to first principles. Now, it's based on water. Now, I want you to understand that most of the initial chemistry was done using water, and that's understandably because we're humans and we're water-based in carbon, right? So, but it turns out that water, if you look at my notes, I have an equation. Water isn't stable. It actually breaks down into hydrogen and hydroxide. Technically, hydrogen and hydronium, but that's a chemistry thing. But it breaks down, not a lot, but it does. So if you have pure water, when it's neutral, and you've got pure water, at any one time, you have 1 times 10 to the negative 7 moles of hydrogen and 1 times 10 to the negative 7 moles of hydroxide, okay? There's your 7, okay? You can see it in my notes. I've got 1 times 10 to the negative 7, and that should already clue to you. Where have I seen the 7? Well, here's the 7, neutral. And I just said that when you have neutral, pure water, you have 1 times 10 to the negative 7 moles of hydrogen and 1 times 10 to the negative 7 moles of hydroxide. So hydrogen and hydroxide. Equal concentrations, negative 7. How do we get rid of this crazy beast, right? Now, this is what this crazy thing is here, okay? So notice that the pH scale, when you go from 0 to 1, 1 to 2, 2 to 3, you're going up by an exponent, okay? 1, 2, 3, 4, 5, all right? So that means that each of these is 10 times different than the other, all right? Not 1 times, 10 times different from its neighbor, okay? Now, if we wanted to simplify that chemistry in math, or when we simplify that math in chemistry, what you do is you take the log of that sucker, okay? So they took the log of that hydrogen ion concentration and they got rid of this 10, and then you were left with negative 6 or negative 7 or negative 8, all right? So it's simpler now to deal with these concentrations, but we still have that annoying negative, all right? Now, what do you do to get rid of a negative in math? Well, to get rid of a negative in math, you multiply something by negative 1. So that's where this crazy thing comes from, the negative log of the hydrogen ion concentration. So if you took the negative log of this, you would be left with 7. Now, ironically though, when you look at the scale, now as you go up, and I'm sure you can tell that 1 times 10 to the negative 6 is more than 1 times 10 to the negative 7, right? But if you take the negative log of that, you get 6, which is a smaller number than 7, all right? So this is where it gets confusing, right? So when you're looking at your pH values, try to keep that straight. Remember, it comes from that negative log. And in the homework, there's a good video goes over this, all right? I really want this not to be a mystery to you, all right? So this is why when things are increasingly acidic, the pH value goes down, all right? Goes down, and increasingly alkaline or basic, the pH value goes up. Okay, because you're looking, it's always based on this hydrogen ion concentration. If it was based on the hydroxide concentration, it would be backwards, okay, from what I just said, all right? But it's not, it's based on the hydrogen ion concentration, and this is why this happens, okay? So I really hope that this makes sense to you, and it's a scale that runs from 0 to 14, all right? Not from 1 to 14. 0 here is a number. It's basically 1 molar, okay? So it's a very concentrated acid, okay? And notice that each of these numbers is 10 times different, okay? Please, all right? So a pH of 0 is super, super acidic. If you look on page 10 of my notes, that's actually 1 molar solution of acid, all right? Now in the body, we have very widely varying pH values, right? So the inside of the small intestine is a pH of 8, it's alkaline or basic. Saliva tends to be a little bit acidic, urine a little bit acidic, varies depending on what you're eating. Gastric juice, pH 2. Now remember, again, that this is not, like if you're talking about pure water and gastric juice, it's not 5 times different, all right? It's 10 times 10 times 10 times 10 times 10 times different, okay? It's a huge difference. It's very, very much more acidic, all right, which is shown here. Right? So if you go from pH 7, all right, down to 2, it's 100,000 times different in pH. Okay, so please, please, please make sure that you understand that. And that's because it's a log scale. Every number is 10 times different from its neighbor. Okay? All right. Now, obviously in the body, we need to protect ourselves from these rapid changes in pH, all right? And the way we do that is we use a molecule called a buffer. And there's all kinds of these, okay? But this molecule has now come back to see us, all right? Here we are, bicarbonate. It is an ion, has a negative charge. And it's kind of a neat molecule because what it does is that it can hide excess acid and hide excess base. not forever, but enough that we can then ship out that excess acid or ship out that excess base from our kidneys, all right? So say you just had a drink of Coca-Cola, which is very acidic, and now you have all that acid circulating in your body, it's in your blood, and you need to get rid of that extra acid through your kidneys, and the way you're going to do that is with your biological buffer, which is bicarbonate, okay? So this is a molecule that you should learn and know well. All right, so this is the chemistry. I'm not going to get you to draw this, but here you can see a bicarbonate can hold on to excess hydrogen or excess base, and then pass it on in the kidneys to protect you. All right. Yes. So yeah, that's what a buffer is. And here's a nice review video on pH. All right. And there's other work in the notes. I mean, the homework. All right. Now, the other thing that you need to be aware of are the different types of chemical reactions, and you should just be able to recognize these. So we have decomposition reactions. So that would be having a molecule and breaking it down into its substances. So that's a decomposing. A synthesis is the opposite. So you're taking things and putting them together. Exchange is when they change partners. All right. So be aware of those and be able to recognize that. Okay. Now, things that affect reaction rates, some of these things should be intuitive. Temperature, you probably know, things speed up. The concentration of your reactants, or the things that are reacting in a reaction, all right? The other things is how reversible it is or how stable the product is. It's a little bit harder to imagine when you're talking about chemistry. But you kind of notice it you look in my notes on page 12. A reversible reaction tends to have the arrows pointing in opposite directions between the reactants and the products. That just means that the products tend to break down. We play with it a little bit more in my chemistry class. And then catalysis. So a catalyst is something that is going to help a reaction take place by lowering what we call the activation energy. All right, you can see this picture in my notes on page 12. So basically, for a reaction to take place, there's a little boost of energy that's needed to have it take place, and a catalyst will lower that and make it easier for a reaction to take place. All right, and in the body, we have really important catalysts that are called enzymes, which is a class of proteins, and almost every reaction in the body relies on them, and we're going to get into that very shortly. Okay. So in metabolism, metabolism, all right, we have catabolic reactions. And if you think back to the other slide that I showed you, those are energy releasing, you're decomposing, and since they're releasing energy, they're called exergonic. Anabolism, you're building things, you're storing energy, they're synthetic, they're pulling in energy, so they're endergonic. We also have what we call redox reactions, which are a combination of an oxidation and a reduction. We're not going to get you to really recognize these. In chemistry, you do a lot of this where electrons are released and energy is released, and reduction, electrons are gained and energy is stored. But those are the main types of reactions that take place in the body. So catabolic, anabolic, oxidation, reduction, and redox, okay? And anabolism and catabolism together make metabolism, all right? Okay, now, the next thing that we get into to finish off this chapter is the major types of biomolecules, all right? Now, the first thing to understand is that the synthesis of these anabolic reactions are dehydration synthesis, which we talked about, and they're broken down by hydrolysis or catabolism, all right? Now, this is basically what we're doing with our food, right? So this is going to remind you what's happening here. 3 types of these major molecules. So the carbohydrates, the proteins, and the nucleic acids. Carbohydrates, proteins, nucleic acids, we say are polymers. So basically what's happening is that you're having, and I'm going to go over this again, subunits that come together, all right, using a dehydration synthesis to form a bond and release water, all right? And these subunits that come together here are called monomers, and they make the polymers. And then when you break down your food, release energy, you're going to then need water to do that. Okay, and then you release the monomers. Lipids are not polymers, but still when you make lipids, they're made through a dehydration synthesis and broken down through a hydrolysis. Okay, and lipids are really good ways of storing energy, but we're going to get into this in a sec. All right. Now, the major molecules are all based on carbon. And the reason in biology, in human biology, carbon is so important is because carbon is really promiscuous. It has 4 valence electrons, so it's right in the middle. It can gain electrons, give electrons, so it's really, really good at bonding. And it can bond to itself even really well. So you can have carbon molecules that would bond to itself. It's capable of forming double bonds and triple bonds between each other. If you take organic, you'll learn that a single bond is an ene, a double bond is an ene, and a triple bond is an yne. It can form bonds with itself and other molecules such as oxygen, nitrogen, hydrogen, and sulfur, which are the other key ingredients to the different biomolecules. The molecules can vary in length. They can be really, really, really long, as you'll see. They can change positions of double bonds. So both of these have 4 carbons, but notice the double bond is in a different place, 1 butene or 2 butene. They can be branched. They can have ring structures. They can be rings with double bonds in them, right? So really, really versatile. They can be really tiny molecules like methane, or huge, huge things. This is a nanotube, which is important to medicine. But they can be, have all these a kaleidoscope of shapes, all right? So carbon is a very, very interesting molecule in terms of bonding, all right? Now, the next idea, which would now bring into lecture 3, is this idea of functional groups. So what you'll see on some of your biomolecules, and there's a couple of exercises in the homework that try to get you to see these. I'm not expecting you to memorize all of these, but hopefully you'll just get to see them, kind of get used to them. But basically what they are groups that are added to another structure. So the rest of the structure is referred to here as R, okay? The rest of the structure, and this is just some of these functional groups. So basically it's these sort of recognizable type of molecule or additions to a structure that have very important properties in a molecule, right? So here's a big sort of biomolecule, and you can see these functional groups show up in here, okay? And they have different names and different functions, all right? So some of the classic ones are hydroxyl, or we sometimes call it an alcohol group, which is an oxygen bonded to hydrogen. It's polar. A methyl group, which is this carbon with 3 hydrogens on it. It's nonpolar. A carbonyl group is just basically a carbon with a double bond to an oxygen. That's polar. A carboxyl combines the carbonyl with the hydroxyl. So you can see here's a carbonyl and here's a hydroxyl. This is acidic. All right, so this carboxyl group is acidic. An amino group has a nitrogen and 2 hydrogens on it. It's basic. A phosphate group, very, very popular. Here you got your phosphate onto basically 4 oxygens, and one of them is a double bond. All right. The other one that you'll see is this thing called a sulfahydryl. It shows up in proteins. It's polar. This thing's also ionic. You can also lose these and become charged. And so on, right? So these are like some of the main ones that you're going to notice as we go through these major molecules, okay? So now let's go through this. And I remind you that when we talk about these biological molecules, 3 types of them, carbohydrates, proteins, nucleic acids, are polymers. So they're made up of monomer units. And lipids are not a polymer, even though they are formed with a dehydration synthesis. Okay, so let's go through some of these. They're all based on carbon. I always start off with carbs. All right, so carbs are basically sugars. All right, so here we have, and I'm just reminding you again here, a dehydration reaction is when you synthesize your polymer or your lipid, and you're going to add energy, you're going to release water, and you're going to make a longer polymer. And release energy when you're breaking that down by hydrolysis. In order to do that, you're going to pull in water. Okay, so you're splitting water, hydrolysis. And you can see how the water is split here. I like this slide because you can see it quite well. All right, so that's what happened to the split water. And back here, well, this is where the water is coming from. Okay, so really, really important in the body. All right, so let's get into our carbs. Now, carbs, as I mentioned, are polymers. We have a certain number of monomer units that we use in the body, and the monomer units are actually functional on their own in the body, and they're sugars, right? So glucose, fructose, and galactose are 6 carbon sugars that we use, and 5 carbon sugars, deoxyribose and ribose. These we use straight up as a monomer. We don't even need to form a polymer here, all right? But these are called monosaccharides, all right? So these are the monomer units, and these come together to form the polymer. Now, the next step, though, is forming these, what we call disaccharides, which are also functional in the body, right? So here we only put 2 of the monomer units together. The most important ones that we talk about in the body are maltose and sucrose and lactose, all right? Lactose I don't have here, I have it in my notes. So lactose, in my notes on page 15, is made up of glucose and galactose, all right? Sucrose, as you can see here, is made up of a glucose and fructose. And maltose, which is your main milk sugar, is made up of 2 glucose molecules, all right? And we use those as well, all right? Now, the polysaccharides, so that's your big polymer. Polysaccharides means many, 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 many sugars, right? A monosaccharide was 1 sugar, a disaccharide was 2 sugars, polysaccharide, many sugars. Your main polysaccharide as a human being is glycogen. Now, remember, these are all based on glucose. So this, if you're thinking, well, if I eat fructose, well, how come I can't make that into my polysaccharide? Well, your body does. It actually has an enzyme, which we'll talk about shortly, that will convert that fructose to glucose and then put it into the polymer glycogen, all right? So your body works on glucose, and then you make your big long polymer, polysaccharide, which is called glycogen. Of course, we eat plants, so their equivalent is a starch, all right? So plant glycogen is starch, and this is mostly used to store energy. Plants also have another form, and that's cellulose. It's still based on sugar, but I'm going to show you on glucose, but I'm going to show you the difference. Cellulose is a slightly different structure, but we need that as well, because that's, for us, it's fiber, dietary fiber, which is really, really important to the body. And you learn about that in AMP2. Now, the differences between starch and glycogen are not much. It's still long chains of glucose molecules, but you'll notice here on this slide that the glycogen is just more branched and more twisted than starch. And starch, if it doesn't have any branches, it's called amylose, and if it has branches and twisted, it's called amylopectin. But really, there isn't that much difference between them, okay? Just that glycogen is more branched. But this is why we can easily use starch, because it's very much like glycogen, all right? And so on. Okay, so hopefully that makes some sense. Let me just see where I'm at here. Okay, now, oh yes, so the other one is cellulose. So cellulose is important for plants because they don't have skeletons, and they use cellulose as structure, right? And we use it as fiber. But why is it, even though it's based on glucose, why is it that we can't break it down like we can glycogen? Now, what I want you to see here, and this is like getting a bit of chemistry, but it's still glucose. But what's happening here is that it's an isomer or a different form of glucose that goes into cellulose. And it's a really, really small change, all right? So glucose, when it's not in solution, is a linear molecule. And then when it goes into solution, it becomes a circular molecule. But the only difference between the glucose that we can digest, which is called alpha glucose, versus the one we can't, which is beta glucose, is that this hydroxyl, I'm using the term, this hydroxyl and this hydrogen, are flipped on this structure. And there is a side in this, because notice there's the last carbon is hanging out here, okay? So when the hydroxyl is on the same side as this methyl group with an hydroxyl on it, we can't digest it. And it's used to make cellulose, all right? If it's the other way around, we can digest it. It makes a huge difference because we have an enzyme that will break down a molecule made up of these, but we do not have the enzyme that will break down these, all right? So here you can sort of see it. So the one that we can digest, notice all the OH groups on the one side, the hydroxyl groups. Whereas in cellulose, they bounce back and forth. And we just don't have the enzyme to do this, right? And also notice that this angle is different, right? So enzymes are very, very important, very, very specific to their reactions. We're going to get into what an enzyme is very, very shortly, okay? But remember, it's a biological catalyst that's needed to perform a chemical reaction, or to ease a chemical reaction. If we don't have the enzyme, we can't do it. Cows eat grass. They don't have the enzyme either, but they have bacteria in their guts that do have the enzyme that's used here. Same thing for termites, that kind of thing. All right, so you need to have the enzyme to break down the cellulose, which we don't have, okay? So that's why when we have beta glucose fiber, this makes up dietary fiber, but even though we can't digest it, is important for our digestion, which sounds crazy, but it is, okay? So again, when you get into nutrition, you'll get to talk about that, all right? Now, lipids. Okay, so lipids, as I said before, are not polymers, even though they look like it. Look at this long chain of carbons. Looks like a polymer. No, it's not. This is a molecule, all right? This is a nonpolar molecule called a fatty acid. So this is why often students get this really confused, because it looks like it should be a polymer. It's not. This is one molecule, fatty acid, all right? Now, lipids, all right, they're made up of different classes of lipids, and usually what we're thinking of when we say fat, we're thinking of this one, the triglyceride, all right? So triglyceride, also known as fats or oils, depending, okay? So what is that? Well, it's made up of a molecule, a 3-carbon molecule called glycerol, and there's a dehydration synthesis to not just one fatty acid, but 3 of them, okay? So if you have glycerol and 3 fatty acids, you have a triglyceride or a fat, okay? Now, I want you to understand that those lipids here, I mean, not these lipids, the fatty acids here in your triglyceride, they don't have to be the same. They can be different, and there's different names for these, all right? I don't know the names offhand. I'm not an organic chemist. I can't just name them like that. But these will have different names, and there's something acid. Like one of them would be palmitic acid or taric acid, all right? So there are these different acids. And they actually have a carbonyl group, carboxyl group on the end. Oh, this is a long video. And it forms an ester linkage actually to the glycerol. All right, so there's dehydration synthesis that takes place here. And these 3 fatty acids are connected to our glycerol, and you have a triglyceride. Sometimes you'll see it on an ingredient list saying triacylglycerol. It's just fat. Okay, so this is what we think of as dietary fat. Okay, now the thing is, remember you've got carbon chains here. Remember sometimes it can form double bonds. All right, so if all of your fatty acid chains have single bonds, we say that they are saturated. All right, so they pack really well. And they're solids at room temperature, like butter. This would be like animal fat. But if we have double bonds in them, they'll form little kinks, so they won't solidify at room temperature. They'll form an oil, right? So the more unsaturated, the more fluid or liquidy it is. And you've got an oil. These tend to be plant fats, all right? So they're more like this, and we consider them healthier. Hopefully that makes sense. So all saturated in fats, all single bond saturated fats, unsaturated fats, or oils. Now, the next type of lipid are the phospholipids, which are hugely important. They are used to form cell membranes, which we'll talk about in chapter 3. They're different. They're very similar to a triglyceride, but notice there's only 2 fatty acid chains. Position 3 is we put in what we call a phosphate group. It's a little bit more than a phosphate. So here's your phosphate, your phosphorus, your 4 oxygens. Remember that from our functional group slide. Okay, going back here. Functional group slide, might as well. Oh, that's so far away. There it is, phosphate group. There it is. Remember when we did the triglycerides? Here's your carboxyl group. Okay. Now let's go back. Okay, there we are. All right, so this thing here has got a charge on it. This thing is hydrophilic. It actually likes to face water. It's water-loving end. But on this side, it's a hydrophobic end, right? These things are nonpolar. It doesn't dissolve well in water. It's oil. So this molecule has very specific properties because one side is hydrophilic, the other side is hydrophobic. We call it amphipathic, okay, amphipathic. And because of this, it's really good at making membranes, right? Because these things here, these hydrophobic tails, will spontaneously organize with other hydrophobic tails. And the hydrophilic head group is going to arrange with water, okay? And then connecting all of that then is our glycerol molecule here. There's the 3 carbons, okay? And this is why these are really important in making cell membranes, right? Because they'll do this spontaneously. No energy required. They will do it on their own simply because of hydrophobicity and hydrophilicity, right? Now, the third classical lipid are the steroids. Now, the steroids are based on cholesterol. They are derived from cholesterol, and that's why cholesterol is actually highlighted here in the yellow. You've got 3 6-member rings bonded to 1 5-membered ring, always in that orientation. That's cholesterol. Now, if you add some functional groups to that, notice you have a hydroxyl, a methyl, a chain with methyls on it. This is going to now be a steroid, all right? So this is the third class of steroids, which are based on cholesterol. So yes, you need cholesterol in your diet, okay? And the thing that makes different steroids different from each other are the functional groups, all right? So what I know will happen when you start doing the endocrine system next semester, you're gonna see the differences between testosterone and estrogen. And if you took 105 with me, I would have shown you that. And the only thing that's different are the functional groups. But remember, testosterone, male hormone, not that females don't have it, but the primary male hormone, and estrogen, not that males don't have it, but the primary female hormone, are still steroids, okay? Very, very similar, only different by the functional groups, okay? So that's your third class of lipid. The other class that is listed in this course are the eicosanoids, all right? Now this is kind of weird, all right, because they are derived from a polyunsaturated phospholipid, okay? So it's not that they're completely different than the other lipids, and really I don't usually see it as a class of lipids, but in this course it's listed there in your learning outcomes. So eicosanoids is a particular type of a phospholipid called an arachidonic acid. And some chemistry happens to this, and it makes some very important classes of molecules that are hugely important in medicine, all right? They're really important in terms of, did I list it here? Let me just see if I listed it. They're really important for signaling in the body or how the body communicates with each other, really important for inflammation. A lot of drugs are targeting these eicosanoids. The 3 main classes are the cyclooxygenases, which are either prostaglandins or thromboxanes, okay? And the lipoxygenases, and of those, the leukotrienes, right? So in my notes, I have prostaglandins, thromboxanes, and leukotrienes. And we mention them because, there's some really complicated chemistry, as you can imagine, that's happening here. But so important to medicine, all right? Because a lot of drugs are working on these, particularly anti-inflammatories, okay? So this is why we want you to be aware of these. And they are lipids in that they're based on these polyunsaturated phospholipids. But don't be surprised if you Google lipids and you don't find eicosanoids. We introduced them here because a lot of you are going into nursing, all right? And you'll learn about it when you take pharmacology, absolutely, okay? Okay. Let me just see. All right, now, cholesterol, the good and the bad and the ugly, all right? So now when they're doing your blood cholesterol levels, they're not really doing cholesterol per se. They're looking at what we call high density lipoprotein and low density lipoprotein, okay? What does that mean? Well, because fat or cholesterol, you need it for your cell membranes, which we'll talk about. But it's hydrophobic, and your blood is hydrophilic, right? And it needs to be transported. So the way the body transports fats like cholesterol, steroids, is in a mixture of lipid and protein, all right? Sort of like a magical structure, I know, so I'm being a little sarcastic. It's not magic. And there's some that have more protein to lipid, so that would be the high density. And others are different. So you have a high ratio of lipid to protein. So you have more fat than protein in the low density lipoprotein, okay? And they carry around your cholesterol, all right? Now, if you have a lot of HDL, this is the one that's considered the good cholesterol, it's indicative of helping cardiovascular disease. And if you have a lot of LDL, that's indicative of contributing to cardiovascular disease, all right? So this comes to this whole idea of atherosclerosis and arteriosclerosis, and you'll learn a bit more about that in lab. But basically, this is the biology that's taking place here, and I want to introduce it. You're going to learn more about it when you do the digestive system. But basically, when you digest fats don't go into the bloodstream directly. The fats are combined into another mixture of protein and lipid called a chylomicron. And it gets absorbed by the gut and eventually ends up in the blood, right? And this then travels to the liver. So the liver then processes these chylomicrons and then sends out what we call very low density lipoprotein, okay? And these are really important for the uptake of fatty acids into the cells. We've got receptors on our cells that pull out the fat that's needed. And what will happen is that eventually these VLDLs will be converted to LDLs, which is what your low density lipoprotein or your bad cholesterol is, okay? So if you eat way too much fat in your diet, you're gonna have more fat than the body needs, and the excess is just gonna be floating around the bloodstream as LDL, okay? Now the liver pumps out HDL or high density lipoprotein, all right? And its job, I'm doing bunny ear quotes with my fingers, you can't see me, but its job is to collect that extra cholesterol or collect that LDL and then send it back to the liver to be processed and eliminated, okay? So if you have too much LDL, the HDLs will not be able to neutralize them all. So they'll build up in the body. Okay, so this is the problem, all right? And what happens, we talk more about it in lab, but basically that extra LDL just gets stuck places like inside your blood vessels, and it starts to occlude the blood vessels by the formation of plaques. Those plaques raise blood pressure because the space in the vessel is compromised. Pieces of that can get dislodged and cause emboli that can get lodged in the brain, cause a stroke, get lodged in the heart, cause a heart attack, and so on, okay? So basically, this is why they're looking at your ratio of HDL to LDL in the body when they're doing a blood test, all right? And that's why the LDL is considered bad cholesterol, but it's more than cholesterol, and the HDL is considered good, all right? So you want to have more HDL than LDL, all right? Okay. All right, moving on. Biological molecule number 3 are the proteins. The proteins are polymers. They are polymers made up of amino acids. The monomer units are amino acids. There are 20 different types of them. The amino acids have an amino end and a carboxyl end. So the amino end is basic and the carboxyl end is acidic. So here you can see the basis of an amino acid. And this goes back to our functional groups: amino end, carboxyl end, joined to a single carbon. Off this carbon is always a hydrogen group. So this is always the backbone. And off of that is an R group. Now the R group is going to be different. We've got 20 different variations of that. So the different amino acids have different R groups. When the amino acids are joined to each other with a dehydration synthesis, they form a special bond called a peptide bond. All right, we want you to learn that term. You have peptide bonds in proteins. This course doesn't explain to you the special nature of the peptide bond. If you want to know what it is, please come see me, because I am a biochemist and I could talk your ear off on that. Okay, well, within reason. It would probably take me 5 minutes, but if you want to know, I will tell you. So it forms a peptide bond. And then these R groups are what make these special. All right, so here's the formation of your peptide bond. So you can see here's 3 different amino acids. Here's your dehydration synthesis. formed a peptide bond, and now you have a three amino acids joined together. Here, this repeating unit is called the backbone. You can see it alternates amino carboxyl, amino carboxyl, amino carboxyl. The carboxyl end is going to be on one side of the protein, the amino end on the other. All right. Now, R groups. Here you can see some R groups. You can see they look very different from each other. Yes, I hope. See, here you have a thiol group. Here you have an alcohol. Here you have a methyl group attached to a sulfur. Very different types of properties. All right. So here you can see all of the amino acids. And don't be scared. I don't expect you to memorize these. But I want you to appreciate that these R groups are different, right? They have different properties. And that affects what we call the folding of a protein or the structure of a protein, okay? So if you have these types of R groups, and you can see the name of the amino acids, if you become a biochemist, you learn these. You can see that these ones are nonpolar. And if they're nonpolar, they don't like water, so they tend to be away from water, and they often are inside of a protein, okay? Then you have hydrophilic. Here you can see that they're polar. Notice there's like, you know, you can form hydrogen bonding here, right? So these guys are polar. They're hydrophilic. So since they do like water, they tend to be on the outside of a protein, all right? Or if there's a protein that has a pore through it, which we'll talk about later. So outside, and then you can have charged ones, so negatively charged and positively charged. And positively charged tend to be basic. The negatively charged tend to be acidic, all right? Meaning that they'll increase the amount of hydrogen in a solution, all right? So, you know, anyways. These also tend to be on the outside because they're charged. They'll interact with water, okay? Now, this will affect the folding. So this is the thing. You have 4 levels of protein folding, all right? And, you know, I'm going to qualify this. I'm going to say polypeptide, okay? Because that will help lower the confusion. When I learned this, they were just called proteins, but I noticed that the modern way of doing this is to call it polypeptide, all right? So when you're forming a polypeptide, meaning you've got many peptide bonds, you're going to fold these molecules. And what we call the primary structure, all that is the sequence of the amino acids. And these can be thousands of amino acids long, right? So this is a relatively small polypeptide. There's what, 127 amino acids. But understand, you can have amino acids strung together by the thousands, right? The protein that's being made here is transthyretin, all right? And notice there's an amino end and a carboxyl end, so there's a negative charge and a positive charge on the side. And then you've got all your amino acids, and they have their functional groups hanging off of them. But the functional groups are not important for the primary structure, which is just the amino acid sequence stabilized by peptide bonds, okay? Secondary structure, what ends up happening is that some bonding, hydrogen bonding specifically, is taking place between the groups of the backbone, not the R groups, of the backbone. So what do I mean? What do I mean, what does Dr. Bohr mean by that? Well, notice you have carbonyl groups, you've got amine groups. These will hydrogen bond with each other. All right, because remember, this is kind of hard to see with only 3 amino acids, but if you have thousands of these backbone molecules can interact, and they'll form hydrogen bonds, and that's what's responsible for the secondary structure. And sometimes you can get very specific kinds of shapes with that. You can have coils, which are called alpha helices, or like flat parts, which are called sheets, okay? And if you look in my notes, I have anti-parallel and parallel pleated sheets, all right? So you can look at the picture there. I'm not expecting you to tell the difference, but you might see that, all right? But these would be secondary structure. So if you see the term alpha helix or beta pleated sheet or pleated sheets or beta sheets, that would be talking about secondary structure existing with hydrogen bonding between atoms in the backbone. Now the tertiary structure of the polypeptide, this is where your R groups are coming into play. You have all of your different types of bonding that we had: hydrogen bonding, hydrophobic interactions, ionic interactions, polar interactions. Here you can even see you can have covalent bonding. This is all stabilizing the tertiary structure, and it's always between the R groups, okay? And this is what you need for your final structure of your polypeptide, okay? So here you have a transthyretin polypeptide. Okay, Now, I said to you there's 4 levels. There is a quaternary structure, but not all proteins require it, okay? If you have a polypeptide or a protein which is now functional as a single unit, all right, you don't need a quaternary structure. This might be the completed protein. However, there are some proteins in the body that function as machines, and you have more than one polypeptide that comes together to form the final protein structure, and that's when we have a quaternary structure, all right? So here you can see that transthyretin is actually one of those. It takes 4 of these units here in the tertiary structure to come together to make the final functional protein, okay? So transthyretin has a quaternary structure. Another example would be hemoglobin. Hemoglobin has 2 alpha proteins or peptides and 2 betas that come together to form hemoglobin molecule, all right? So hemoglobin would also have a quaternary structure, all right? So here's your summary. So primary structure, amino acid sequence. Secondary structure is hydrogen bonding between atoms of the backbone. Tertiary and quaternary structure are R group related, and tertiary is the folding of the polypeptide, single one. Quaternary structure, not always needed, but if more than one subunit comes together, stabilized by R groups, all right? Interactions, of which could be any of our bonding types that we discussed earlier in this very, very long video, okay? Now, when you denature proteins, you know, this is huge, right? So if you're cooking an egg, you crack your egg, you heat it, and then it turns white, right? The white of the egg turns white. And the reason that happens is because you've destroyed the protein structure and you've denatured it. Now, all this means is that the protein has been unfolded, okay? Denaturation is the unfolding of a protein. It does not affect primary structure. You actually have to do severe chemical reactions to break down the primary structure, all right? Those peptide bonds are pretty strong, all right? So what's happening when you denature a protein is that you're removing the quaternary, tertiary, and secondary structure. Primary structure remains intact. So that's why we call it the unfolding of the protein. Don't ever say that you've destroyed the protein. In certain situations, you can refold it, okay? Now, how can you do that? Well, changes in pH. pH incredibly important to the folding of a protein because it changes the charges on the R groups, okay? So really, really important. So pH, huge. So remember when I showed you all of the different pHs in the body, if you have different compartments in the body that have different pHs, you need to have different proteins in those different compartments, all right? So you'll learn about that when you do the digestive system. Salt concentration will also change these charges, right? Because you know now, salts are electrolytes. They have charges. They will affect the charges on the R groups, okay? And this is why in the old times, when they would want to preserve food, they would just throw preservative, but use salt, right? So you can kill things by putting the salt in there, kill the bacteria, and preserve the food, okay? Temperature, that's just like I just said, cooking, right? Temperature will also unfold. And then the other one is the presence of metals, all right? Now, the best way for me to have this importance of this be related to you is, I'm sure you know about the Flint water crisis, right? And they had metals in the water, right? And this is so important for human development of the brain, because if you've got those metals in there, the proteins aren't folding properly, and that's going to be really, really bad for development, okay? So presence of metals will also cause denaturation, all right? Okay. I haven't even been looking at my notes. I've been doing this for years. Okay, now, now enzymes, going back to that, remember these are hugely important because they are going to be responsible for most of the chemistry in the body. Because in the body, we can't just add heat, right? We can't take a person and raise their temperature to 140 for a chemical reaction to take place. Can't do it, right? So we have to lower the amount of energy that's required for a reaction to take place. We use a catalyst to do that. And our catalysts are enzymes. Enzymes are proteins, all right? Because enzymes are proteins, they can be denatured by all of these means, okay? So this is why we have to maintain homeostasis so that we don't denature our proteins, okay? So remember, what are they doing? They're lowering the activation energy, all right, releases energy in small steps. These enzymes, because they're proteins, they're coded for in our DNA, all right, they're very, very specific for the reactions that they take place. Think back to what I was saying between glycogen and cellulose. Even though it's a small little change to that glucose molecule, we can't digest cellulose. We don't have the enzyme, all right? They may require a cofactor or another little molecule in there to help them maintain their structure. That can be inorganic or organic, all right? So if you're talking about hemoglobin, it's both, right? So hemoglobin has a heme group, which is organic. It's inside of the sections of your hemoglobin molecule. It's stabilized there. And the inorganic component is the iron. The iron is holding onto the oxygen in the hemoglobin, which is sitting inside this heme group, all right? So you can see pictures of hemoglobin, you'll see what I mean, all right? So that's not an enzyme, it's a protein, but it's a good way to think about it, right? So a lot of enzymes also need cofactors to function, all right? I have a series of enzymes that I study in the lab called metalloproteinases. They need a metal, so it's an inorganic cofactor in order for them to function, all right? So how does this work? So what happens is that enzyme, so this is sucrase, this is an enzyme that's gonna break down sucrose, right? And here's your bond here. And there's an active site in the enzyme which interacts with the substrate, which is sucrose. Then it's going to do things to that bond to enable the bond to break, all right? Sometimes it stretches it or twists it, or it just stabilizes it so that it can break, all right? And then out will come your products, which are glucose and fructose. All right, but notice that the enzyme itself, at the end of it, it might change its shape a little bit in the process of doing its job, but at the end of it, the enzyme comes back out intact and it can be recycled and do the same thing over and over and over and over again. So catalyst lowers the activation energy for a reaction, but does not get consumed by that reaction, okay? All right, so what can we use to affect an enzyme's function? Well, pH and temperature, very, very critical in the human body, right? That's why, you're at 37 degrees centigrade. That's what our enzymes like. Your pH is going to be dependent on your compartment. But in general, we think of the human body as being around pH 7.4, all right? Some places a little higher or lower, but blood pH around 7.4. So we think of body pH being around there. And remember, the difference between 7 and 8 is 10 times, right? So a 7.4 is not just a fraction of a percentage, right? All right, so it's a lot, all right? But that's usually what we work with, and so on, right? And different organisms use different temperatures. My planarian in the lab, they're not at 37, they're at 20 degrees, right? So their enzymes function at a different temperature, all right? Okay, now the last type of biological molecule are the nucleotides. ATP, nucleotides, and nucleic acids, all right? So ATP is an energy transfer molecule, all right? I know you're gonna say it's an energy storage molecule. It's very, very, very confusing, all right? So I'm not gonna be really upset with you if you say that it's an energy storage molecule, but it's not really. Our energy storage molecules are actually glycogen, and so fat fat and our big carbs, right? That's really the storage molecule. ATP is transferring the energy technically, all right? And I know if you're taking AMP2, that will be with Dr. Tice, and she's going to want to make sure you know that, all right? So it's transferring the energy from one place to another, and you should go over, if you can't remember, glycolysis, and a Krebs cycle and oxidative phosphorylation, please, please, please go over it. I mean, obviously this is a lot already, but I have a video here to help you refresh your memory as to how that works. I know that Dr. Tice will expect you to remember that in AMP2. So here's this video. It's very convenient. Have a look as to how much you need to know. Use your study guide, all right? So I'm not, this is a lot on its own, all right? Other molecules that we can use are GTP, which is also able to transfer energy. We have cyclic AMP, which I will talk about in the chapter where we're talking about cell membranes, and that is chapter 3. But it's a second messenger, but it's also a type of nucleotide. And then of course our nucleic acids, right? So that's what I'm just gonna remind you of. So remember ATP, there's your phosphate groups. It's a triphosphate. And again, there's your functional groups. This one is considered the high energy bond here. So basically ATP is going to transfer that energy in this high energy bond. You're going to hydrolyze that, all right? So it's hydrolysis. You're going to split water, all right, to do this. You're going to pop off your phosphate. You're going to have ADP. Then you can store it back up again. Put energy back in, you put the phosphate back on, all right? So this is recyclable. This energy transfer molecule is recyclable. But this is what we call our energy currency, right? So we need to have ATP so we can function. And so that energy from our food is going to be transferred. How do we extract it from our food? Well, review this, right? So remember, there's aerobic and anaerobic metabolism. Please review it, okay? All right. Now our nucleic acids. So this is DNA and RNA, all right? So polymer of nucleotides, all right? So each nucleotide contains a phosphate group bonded to the 5th carbon of a 5-carbon sugar, which is your pentose, and a base, okay? So here's your base. And the base is attached to carbon 1. Carbon 3 is highlighted because there's 2 different pentose sugars that we use, and I'm going to show you that in the next slide, all right? But basically, this is our nucleotide. Without the phosphate group, this thing is called a nucleoside. You might see that, all right? So they are our building blocks. There's 5 different ones, all right, between DNA and RNA. Here they are, all right? So, and they vary by the bases. So the 6-carbon base, or the 6-member ring bases, they're not carbons, nitrogens in there too, are called pyrimidines, of which we've got 3. And then we have the purines, which are a 5-carbon ring connected to a 6-carbon ring. Those are, like I said, the purines. And we've got 2 of those. So there's cytosine, thymine, uracil, adenine, and guanine, all right? For me, I'm just happy if you remember C, T, U, G, and A, okay? Now, the thymine only in DNA, not in RNA. The uracil replaces the thymine in RNA, all right? So then when we're also looking at our sugars, so DNA is deoxyribonucleic acid, so that's where the sugar is different. RNA is just ribonucleic acid. So what's the difference? Well, look at carbon 2. There's either a hydrogen here for deoxyribose, or there's a hydroxyl group here in ribose. Okay, so that's the difference. So this is an RNA, this is in DNA. Okay, now these things bond to each other. Oops, I don't even have that there. So that's in my notes. Let's go back. Popped right out of my thing here. Let's go down here. Hello, where are you? Okay, so in my notes, let me go here. I have a table that compares DNA to RNA. All right, so this is on page 23, right at the end here. So in DNA, you've got a double-stranded helix, and in RNA, you only have a single strand, all right? In DNA, you have a deoxyribose, and ribose is only in RNA. G always pairs with C, whether you're in DNA or RNA. In DNA, A pairs with T, and in RNA, A pairs with U. DNA, like I said, is a double-stranded helix. If you look in my notes, you'll see that it is anti-parallel. What does that mean? Okay, this is what I mean. Notice on your backbone of your nucleic acid, you have a 5' end from your phosphate, when the phosphate is bound, and the 3' end where your hydroxyl is, okay? When you have DNA, they're anti-parallel. So the other strand here, on this end, it would be the 5', and over here would be 3'. That's what we mean by anti-parallel. All right, now when you're dealing with the ribose, it just folds on itself, all right? But we have 3 different types, okay? We have messenger RNA, ribosomal RNA, and transfer RNA, okay? And this is something that, and I've noticed in most A&P courses, A&P1 courses, they go over this part of it. And in this course, we don't, okay? So if you want to see a review on the different types of RNA, I've got this video link here, all right? So hopefully it works. So it's the types of RNA. So hopefully that will refresh your memory, all right? And other than that, this has been a lot, okay? And I think that will be enough to get you through. Now, remember, again, this is a huge, huge chapter, and I understand that. So don't panic per se. Look at the study guide, use it to ground you so that you're ready for the test, because it's not just this on the first test, right? But this is really supposed to be review, all right? And I believe me, I know it's not, because it takes years to learn all this stuff, all right? So I just want you to try your best. That's all I can expect. And believe me, if you have problems, please come and see me, okay? Or reach out to me with Teams, or arrange a meeting with me somehow, and I will help you, okay? Please don't panic. Panicking is not going to be helpful, all right? But chemistry is important to A&P, right? And that will become more and more apparent as you go on in your studies, not just here, but as you keep going, all right? So just trust me, all right? Trust me, it's important, all right? I know it's hard and I know it's a lot, all right? But I'm in your corner and I want to help, all right? Okay, so that's it. It's a really long video, and hopefully everything is going to come and gel for you enough that you can do well on the test, all right?